Nucleic acids are one of the four important biomolecules that are found in the human body. They are also organic molecules. They are also considered organic, and they are also a class of macromolecules. The building blocks for nucleic acids are nucleotides. So nucleotides can be considered as the building blocks or the monomers that help to form the nucleic acids. From the name, we can deduce that nucleic acids are acidic molecules that are located in the nucleus. Nucleic refers to the nucleus, where acids refer to their acidic property. The two important nucleic acids include DNA and RNA, deoxyribonucleic acid and ribonucleic acid. So in this video, we will be discussing the structure and important functions of nucleic acids. So DNA stands for deoxyribonucleic acid. So it is the genetic material that is found in all the organisms, from the single-celled bacteria to all the complex multicellular organisms. In eukaryotes, DNA is found in the nucleus of the cells. Other than nucleus, DNA can also be found in the mitochondria and chloroplasts. So in the case of prokaryotes, which include bacteria, the DNA is not surrounded by a nuclear membrane and it lies freely in the cytoplasm of the bacterial cell. So this is the most fundamental difference. In the case of eukaryotes, the DNA is surrounded by a nuclear envelope and is contained in a nucleus, whereas it resides freely in the cytoplasm of prokaryotes. In eukaryotic cells, DNA also exists in combination with a group of proteins that are called histones. So histone proteins are basic in nature and they contain a high proportion of positive charges. While DNA, on the other hand, is usually negatively charged. So we'll discuss this shortly. So DNA is negatively charged because of the phosphate group that is present in its structure. So we have negatively charged DNA that is interacting with the positively charged histone proteins. So these opposite charges allow them to combine with each other, and this combination is actually considered as chromatin. The association of negatively charged DNA with the positively charged histone molecules together comprise the chromatin. So histones help to wrap the DNA inside the nucleus. Although most organisms in nature contain DNA as the major source of genetic information, we also do have some viruses that actually contain RNA as the major source of genetic information. So these organisms or these viruses actually have RNA instead of DNA as the major source of genetic information. All the cells present in the human body in different tissues contain the same amount of DNA. If we were to take out the DNA that is present in the cells and stretch it out, then it would measure 2 meters in length. So this is the total amount of DNA that is present in one single cell. And if we see the average size of an animal cell, it is usually only around 5 to 10 microns or 5 to 10 micrometers. A single animal cell itself measures only 5 to 10 microns, and it is able to hold 2 meters of DNA within its nucleus. So for the DNA to actually fit inside this tiny nucleus, the DNA has to be extensively coiled, and this requires the presence of basic proteins named histones. So histones are absolutely essential for the coiling of DNA, and this extensive coiling ensures that the DNA can fit inside the nucleus. The DNA which is present in the body cells is actually distributed among 46 chromosomes. So all the cells in the human body contain their DNA within the 46 chromosomes. So chromosomes are thread-like structures that become evident during the division of cells or during cell division. So nucleotides are the building blocks or monomeric units that help to form the nucleic acids, DNA and RNA. A nucleotide has three important components. It has a 5-carbon sugar, a nitrogen containing base and a phosphate group. This is the basic structure of a nucleotide. This is the 5-carbon sugar. Then we have a nitrogen containing base and a phosphate group on the other end. So there are basically two different types of sugars. We have a ribose sugar and the other one is deoxyribose. If you look at the molecular formulas of these sugars, you will notice that they are almost identical except for one oxygen in deoxyribose. Now deoxyribose sugar is present in DNA whereas ribose sugar is present in RNA. So deoxyribose sugar just contains one oxygen less than that of the ribose sugar. And this is very important because this actually makes DNA more stable than RNA. And this is usually seen at this position. If this position contains OH group, then it is considered RNA. Whereas DNA only contains a hydrogen group at this location. Next we have the nitrogenous bases or nitrogen containing base, which is also one important component of nucleotide. So they are basically divided in two categories. We have the purines and we have the pyrimidines. So nitrogenous bases are also organic molecules that are made up of the atoms carbon and nitrogen. It is considered a base because they contain an amino group in their structure and this amino group can accept H plus ions. So because they have an amino group that is able to accept protons, they are considered as bases. So coming to purines, purines are made up of two rings. So they have a 6 plus 5 membered ring that is common to the structure of purines. So we have two different purines, adenine and guanine. So these are the two purines that are found in DNA. Pyrimidines on the other hand are made up of a single 6-membered ring. We have three different pyrimidines, cytosine, thymine and uracil. Uracil is a nitrogenous base that is unique to RNA. Uracil is not observed in DNA because uracil's position is occupied by thymine. In the structure of DNA, purine is always hydrogen bonded to a pyrimidine. So adenine will hydrogen bond to thymine. Adenine is a purine and thymine is a pyrimidine. Similarly, guanine, which is a purine, is hydrogen bonded to cytosine. So the nitrogenous bases on both the chains of DNA are held together by hydrogen bonds that help to hold the two chains together. Always remember, a purine can only hydrogen bond to another pyrimidine, or a pyrimidine will hydrogen bond with a purine. So the last component of a nucleotide is a phosphate group. So phosphate is usually designated PO4 3-. It is due to the presence of this group that DNA and RNA are acidic in nature. So this can release H plus ions and that's why it is classified as a acid. We all know that acids are proton donors, whereas bases are proton acceptors. So earlier we have seen that because of the amino group present, the amino group can accept the protons, and that's why nitrogenous bases are basic in nature. Here we have a phosphate group or phosphoric acid that is able to donate its protons, and that's why this can act as a acid, and this gives the overall acidic nature to nucleic acids. Apart from DNA, we also see phosphate group in the structure of ATP and also in phospholipids, which are present in the cell membrane of the cell. This is the basic structure of a DNA molecule. A single nucleotide is shown circled on the top. So a nucleotide has three basic components. We have the 5-carbon sugar, we have a phosphate group which is derived from phosphoric acid, and we have the nitrogenous bases, which can be either a purine or a pyrimidine. The phosphate groups, which are labeled pink and shown as a circle, are extremely important because they help to link the two successive nucleotide molecules together. This is called the sugar phosphate backbone. And this backbone forms the structural framework for both DNA and RNA. The hydrogen bonds at the center actually help to hold the purines and pyrimidines together. If there is a purine on one chain, the other chain would have a pyrimidine or vice versa. Now each chain of DNA contains many nucleotides. As there are many molecules of nucleotides in a single chain of DNA, it is also known as a polynucleotide chain. Polynucleotide chain. So poly means many. So many molecules of nucleotide in a single chain of DNA. So as we can see, there are two polynucleotide chains in the structure of DNA. So DNA can be considered as being made up of two polynucleotide chains or strands that twist around each other to form a double helix. So the two polynucleotide chains basically wrap around each other to form what is known as a double helix. Let us look at the major functions of nucleic acids. The most important function of DNA in the cells is to produce proteins. As we know, proteins are an important class of biomolecules and have varied functions in the human body. So the information or the instructions to produce all the proteins in the human body has to come from DNA. So to produce a specific protein, the DNA will always form an intermediate molecule called RNA or ribonucleic acid. This process is called transcription, and this takes place in the nucleus of the cell. So to produce any protein, DNA has to first form an intermediate molecule, which is usually called messenger RNA. M stands for messenger because the RNA is carrying the message for the protein structure. So the conversion of DNA to RNA is called transcription. Messenger RNA that is produced travels to the site of ribosomes because it is at ribosomes that the proteins are synthesized. So the information that is present in mRNA is utilized by ribosomes to form a specific protein, and this process is called translation. So this is how proteins are produced. For every protein, the information has to come from DNA. DNA will first produce an intermediate molecule which is called messenger RNA in a process called transcription. Messenger RNA is then taken to the ribosomes, where ribosomes will use the information to produce a specific protein, and this process is called translation. So this is the most important function of DNA in all the cells of the body or in any other organism. Another important function of DNA is to store the genetic information. Like we discussed, All the instructions that are required by cells to produce specific proteins is located in the DNA. So DNA is a storehouse of all the genetic information that is required by the cells. Next function is the transmission of genetic information. So whenever organisms divide, the new daughter cells or the offspring produced will also get a copy of the parental DNA. In any form of reproduction, it is important to ensure that the new cells produced also have the same DNA complement as the parent organism, because DNA carries the information to produce proteins that control a lot of traits in the human body. So before any cell divides to form two daughter cells, it is important for the whole DNA to be duplicated so that the daughter cells will also receive the same complement of DNA as the parent cell. One important point, most of the plant and animal viruses contain RNA as the major source of genetic information. However, because RNA is less stable, as we discussed, because of its 5-carbon sugar, most of the multicellular organisms evolve to contain DNA as the major source of genetic information.